The overall goal of this procedure is to study molecular scale hydrodynamic processes in liquids using an analog technique that measures single and collective particle dynamics in vibrated grain piles.
The main advantage of this technique is that it provides an experimentally accessible inverse microscope for directly observing complex molecular scale dynamics.
Since this technique is strongly coupled in the statistical mechanics of interacting particle systems, it opens a door for experimental investigation into fundamental questions in this field.
This technique can also be applied to study the statistical mechanics of interacting macroscopic particle systems, such as the vibrating grain piles we are investigating.
Generally, individuals using this technique will struggle learning the elements of equilibrium and non-equilibrium statistical mechanics that are necessary for interpreting experimental observations and designing experiments.
Visual demonstration of this technique is critical since it extends particle imaging velocimetry, normally used for measuring the motion of micron-scale particles, to the motion of centimeter-scale grains.
The experiment uses this vibratory system. It consists of an annular polyurethane bowl attached to a single-speed unbalanced motor to generate vibrations. These are attached to a weighted base and separated by a group of springs. Attach the bowl assembly to its stand with rubber hooks. Inside the bowl, attach a triaxial accelerometer. This is connected to a sensor signal on a table away from the vibratory system. Next, work with the chosen media for the experiment. In this case, use a ceramic polishing media straight cut triangle with equal side lengths. Add the media to the vibrator.